Today's topic is about microsporogenesis. First, we'll talk about that. What is sporogenesis? Sporophytes bear spore. See, sporophytes bear spore-producing organ and produce spores by process is called sporogenesis. So, what is sporogenesis? Sporophytes which bears spore-producing organs that and produce spore by the process. That process is called as sporogenesis. After that sporogenesis, microsporogenesis. Microsporogenesis is the process. It is also the formation. See, here the definition is the formation of the microspore or the pollen grain is termed as microsporogenesis. So, this is formation of microspore is known as microsporogenesis. That process is termed as microsporogenesis. And microspore is also called as pollen grain. Okay. Next, it occurs in microsporangium. See, structure. One structure that is, this is the structure. Okay. Here the microspores, and this is called as microsporangium. Means it is a pollen sac. Inside it, what is present? That is pollen grain, and that pollen grain is also known as microspore. Okay, which further helps in the pollination. Okay, and formation of new when it get fertilizes with the female gametophyte, and then it's form a new seed or formation of new flower. Okay. Next, anther is a fertile structure of stamen. Anther is a fertile structure of a stamen. A typical anther is tetrasporangiate type. Means a anther is tetrasporangiate type. It has the different forms. There are 4 different kinds of forms of anther, that is isobilateral, decussate, tetrasporangiate, and linear. This kind of microspore are found, but typically mostly we can see that the tetrasporangiate type. Each anther lobe has a 2 microsporangia. Means each anther lobe has a 2. Suppose that this is the anther lobe, so each has a 2 microsporangium or a pollen sac. In each lobe, some hypodermal cells become more prominent and constituent the archisporium. The archisporial cell divides periclinally to form a parietal layer to outer the parietal layer to the outer side, and the inner is called sporogenous cell. Suppose that we are giving the diagram. Sure, this is the stamen. Okay, and this is the anther. Okay, this whole is a anther structure, and inside the anther there is a pollen sac. This is a pollen sac, and inside these are the pollen grains. Okay, we can see one structure out of this. One structure of anther is over here. This is a bilobed structure. Okay, and 4 pollen sacs are found, and in this pollen sac there is a pollen grains. So in this one we can put it out over here. This is what? This is a microsporangium. Inside that the microspore, and this microspore are arranged in a tetrad form. Okay, that we called as over here the tetrasporangiate type. So this is a tetrasporangiate type. Now we see that this is a microspore. This microspore get divide. Okay, this get divided by the meiosis. What happens over here? Meiosis. And in meiosis, what happened? 2 types of cells are formed. First cell we called as the vegetative cell, and the second one is called as the generative cells. Okay, and this vegetative and generative cells undergoes the mitosis first. Okay, after mitosis first, it again transform into the mitosis second, and it forms 4 celled. Okay, so here the two are the male gametes, and one is the pollen tube germination formation occur. Next is what? That this layer gets forms. The 2 male gametes are found, and one cell that elongated into the tube cell. Okay, and one tube nucleus is over here. And this is the pollen layer. Okay, this is the pollen layer, and this outer layer is called as the exine. and the inner layer that bears, that is enzyme. This is the pollen structure, and this enzyme comprises of sporopollenin. Sporopollenin. What this sporopollenin do? This sporopollenin mainly protect the pollen grains from the outer any types of pathogen or kind of external injuries by the pathogen, and also it can also help as a preservation, which can preserve the pollen grain for the further use. Next we see the formation of male gamete, how the male gametes are formed. Okay, so first we will see the difference between the microsporogenesis and the microgametogenesis. What is the difference between the microsporogenesis and the gametogenesis? There is a big difference that we see. That formation of microspore, see, this is the microsporogenesis. So what happens? The formation of microspore within the pollen sac is known as microsporogenesis. Where what happens? The microspore which forms within the pollen sac is known as microsporogenesis. And what is microgametogenesis? Microspores divide mitotically, forming two symmetrical cells. Okay, so this is the formation of pollen grain, formation of microspore inside the pollen sac, and this is the division of microspore. Okay, next in the microsporogenesis, these sporogenous cells differentiated into the microsporocyte, means primary mother cell, it get differentiated. And what happened in this? This is the microspore divide mitotically, forming two symmetrical cells. A large cell is called a vegetative cell or a tube cell, or a small cell is called a generative cell. Second, after first mitotic division, it gives rise to the second mitotic division, and in this it produces generative cells. Okay. Second mitotic division produces generative cell produces two non-motile male gamete. What happens? There is the two types of division, first mitosis division and the second mitotic division. In first mitotic division, what it can be said that two types of cells are formed. First one is vegetative cell and the second one is generative cell. The vegetative cell is the tube cell and the generative cell is a small cell. After that, there is the second mitotic division in microgametogenesis, and what happens? The gamete formation takes place. Which types of gamete? This is the non-motile male gamete was formed in the second mitotic division of microgametogenesis. This is the formation of gamete, so that is it can be called as the microgametogenesis. And here is the formation of microspores, so that is it is called as the microsporogenesis. Each diploid primary mother cell give rise to a tetrad of haploid microspore. And which type of microspore are formed? That is haploid microspore. Okay, now there is, I can show that some steps which you must know about that. This is a very simple and easy step. That is one, first archisporial cell, it undergoes the primary sporogenous cell, that is of 2n. This is diploid in stage. The pollen mother cell is also in diploid stage, and this pollen mother cell and the formation of microspore, here is the meiosis takes place, and this process is known as microsporogenesis. After microspore, when the pollen mother cell undergoes the meiosis, the microspore become haploid in nature. This is the significance, that is haploid in nature, it becomes haploid. After haploid, what happens? Two types of mitosis division occur, in the first mitosis and the second mitosis. What we can say that the first mitosis, it becomes to a gametogenesis, and in first mitosis there are two types of cells are formed, the vegetative cells and the generative cells. So, and after that, the second mitotic division, it first give rise to vegetative cell and generative cell. And after the second mitosis division, what? Formation of male gametophyte, and this process is known as microgametogenesis, and this whole sequence is known as microsporogenesis. This is the whole concept of microsporogenesis.